Hello everyone. Welcome to this podcast for genetics. In the past several podcasts, we've talked about replication in bacteria and eukaryotes. In this podcast, I would like to talk about the application of concepts from our knowledge of replication. Often we, as scientists, we learn about the basic biology of cells and of genetics. And then the next step often is how do we apply it to real-world problems. So let's say we had this segment of DNA that contained the gene enolase, one of my favorite enzymes of all time. It's a glycolytic enzyme, if you remember from when you took cell biology. And let's say we wanted to take this piece of DNA that we had isolated from, and it was outside of the cell, and we wanted to make many copies of this gene enolase. How could we go about doing that? There are two general ways that we can copy this gene, any gene. I'm just using enolase as an example, but there are two ways to accomplish this. So how to make DNA copies? The first involves a strategy that we're going to initially just call in vivo. In Latin, in vivo means essentially in a living thing. So everything that happens in our bodies or other organisms' bodies would be considered in vivo. And the other way is called in vitro. Literally, this means in glass. But practically what we mean by this is we're saying it's outside of the body, outside of a living body. So if something's occurring in a test tube, we would say it's in vitro. I want to begin by spending a little bit of time talking about in vivo and then finish up with in vitro. So let's talk about in vivo. And remember, this is in a living organism. The way we're going to mass produce a gene in an in vivo system is by a process that we're going to call gene cloning. We're not talking about cloning a whole organism here, but just a gene. So let's take our enolase gene, and in between these lines is the actual gene enolase, and over here are just extra sequences upstream and downstream of the gene. And when we say upstream, we mean this way of the gene, and downstream means this way of the gene. And remember, right now this gene, for practical purposes, is outside of a living organism. We have this, say, in a test tube. Then we're going to take a plasmid, and a plasmid is, and we've mentioned this before, and we'll come back to it in a couple lessons from now, but a plasmid is a circular piece of DNA that is often found in bacteria. And what we're going to do is we're going to cut open this plasmid, and it's going to open up the plasmid like so. We're also going to cut the enolase in such a way that we're going to make the ends match the ends here. See how I drew these as a bit of an overhang? And so now it's going to fit nice and neatly right in here. So again, here's our enolase gene. And the way, I should mention this real quick, the way that we insert this in here is we actually use that enzyme DNA ligase that you learned about with replication. The same enzyme. We're able to bring these two ends together. I also want to mention one quick thing in case it comes up later in your studies. The way that these pieces of DNA, the plasmid and the enolase gene, are cut is with these enzymes called restriction enzymes. We could spend a day talking about restriction enzymes, but I just want to spend 30 seconds. There are many different kinds of restriction enzymes, and they cut them in the DNA in unique ways. And so if you use the same restriction enzyme on the plasmid that you use on the gene of interest, it'll allow it to efficiently fit right into this plasmid, just as we see here. So we have our gene here in our plasmid. And now what we do is we place this gene into an E. coli cell. You could use other kinds of bacteria, but E. coli is the organism of choice for most of these experiments. Now obviously I drew this out of scale. It's not really bigger than the E. coli cell. So now this plasmid is inside of the E. coli. And inside of the E. coli, it's going to make multiple copies of it. In addition to these extra copies here, that is, every time this E. coli cell divides, it's going to copy this plasmid multiple times. So every time it divides, we end up making more copies of the plasmid. And what we're really interested in is making more copies of the enolase gene. Okay, so we can keep doing this over and over again, but you get the idea. And the reason we call this in vivo again is because this copying of this enolase gene, in this case, or any gene, but remember we're just talking about enolase now, is using the rules of replication. Just like we learned in the past several podcasts, how the DNA is pulled apart and then the primers come in and lay down the primer, and then we make more DNA using the exact same enzymes we've learned about. So we're taking advantage of the fact that E. coli uses essentially the same kinds of enzymes that human cells would use, and we're making multiple copies of this gene that we're interested in studying. So again, we're applying our rules of replication to mass produce a gene we're interested in studying. Okay, now let's move to the in vitro mechanism. All right, so in vitro, and remember we said that means in glass, or for our practical purposes, it means outside the body. And again, remember, we still want to take our gene enolase here, I just put E here for enolase, and make multiple copies of it. And we're going to use a strategy called PCR. And you're going to do this in lab. PCR means polymerase chain reaction. PCR. So there are 3 steps in PCR that will help us accomplish mass producing this gene enolase. Or again, remember, any gene or sequence you're interested in studying. So let's draw our gene here. And so the first step is called denaturation. And what we do during this denaturation step is we heat the DNA over here to about 95 degrees Celsius. And what that does is it pulls apart the DNA. And let's provide the 5' and 3' ends here. So it pulls the DNA apart by ripping through these hydrogen bonds. So as we go through this, I'm going to indicate how it's similar to in vivo replication. So by heating this up, it's taking the place of the enzyme helicase. Remember, helicase pulled the DNA apart. It's expensive to use enzymes, so it's more efficient to just heat it up and pull those DNA strands apart. Okay, let's not forget to put our 5' and 3' ends here, because the next step is going to require some knowledge of why these ends are important. Alright, so this next step is called annealing. And the temperature that annealing happens varies, really anywhere from 35 degrees to 65 degrees Celsius. And so annealing is when primers that we design anneal to the DNA. And it's going to follow the same rules that we had for replication. The primers are going to bind to the 3' end, because remember, DNA, when it's replicated, is read 3' to 5', and the new DNA that is made is laid down 5' to 3'. So this primer here has a 5' end, and I'm going to erase that here, put 5' on the pink one here, and redraw 3' up here. So these primers being added is equivalent to RNA primase making primers. Okay, now this next step, let's go and number these. 1, 2, 3. So our third step here is called elongation. And during elongation, we're going to extend the length of the DNA from these primers. So during this step of elongation, DNA polymerase, similar to the DNA polymerase 3 we learned about with replication, this DNA polymerase will take this primer and extend the length of the DNA. Then what we do, oh, I should also mention that this occurs, depending on the enzyme that we use, anywhere from 60 to 75 degrees Celsius. Then what happens is we repeat denaturation, annealing, elongation, and we usually repeat it about 35 times. So for instance, after the next round of replication, this DNA strand here will separate, and this one down here, the strands will separate during the denaturation step. Annealing occur where the primers are added, and then they'll extend just as we saw during elongation previously. And every time you repeat this, you double this amount. So you can see after 35 times, you would end up with many times the amount of DNA that you started with. So to be consistent, I'm going to write down here, the enzyme is DNA polymerase. Now scientists knew that this process could happen, and they were able to do it in a lab, but it didn't become efficient and widely used until two major discoveries were made, or we might think of two innovations. This is going to get a little crowded here, but as I talk about these two innovations, I want to be able to refer to what is on the diagram here. The first is the discovery of Taq DNA polymerase. Most DNA polymerases that we know about or knew about at that time, if you heated them to 95 degrees, they would denature. So you could do one round of replication, adding the DNA polymerase here, and then you'd have to heat it up again. That would denature, destroy essentially the enzyme, and then once you're down here again, you'd have to add the enzyme again. So every time you repeated this, you would have to add a little bit more enzymes. Now that works, but it's not very cost effective, and it's certainly not very efficient. So the nice thing about Taq DNA polymerase is that it is heat stable, meaning it can survive at 95 degrees Celsius. And in fact, it is optimal at 60 to 75 degrees Celsius. I should mention, before they found this, this wasn't done at this temperature, it was done at 37 degrees. But this allowed us to crank it up to 60 to 75 degrees. But most importantly, it didn't denature at 95 degrees. That way, you didn't have to keep adding more enzyme. You could add enzyme at the very beginning and let it go all 35 cycles, and the enzyme would be fine. It's a very robust enzyme. So why is it heat stable? It's heat stable because it was isolated from a bacterium that was found in Yellowstone Park and their hot springs. So it's very heat stable because of that. And the second innovation is that it became automated. Prior to automating it, you could accomplish this and you would have 3 water baths, one set at 95 degrees, one set at 35 to 65 degrees, and another set at 60 to 75 degrees. And you would just have to take the test tubes from one water bath, transfer them to another water bath, and then the third, and time that and repeat that 35 times. So you had to be there for the entire time. Having it automated using a PCR machine meant that you could put your samples into the PCR machine and just let it go until it was done. It made it much more efficient. Okay, again, I'm going to be a little crowded here, but I guess that's okay, because I may want to refer to this diagram as I talk about some limitations of PCR, some of which we've already been able to overcome. But let's talk about 4 such limitations. The first is that you need to know something about the sequence. You can't just do PCR on a sequence that you know nothing about. And the reason for that is because of these primers. You have to know which primers to use, the sequence of these primers, so that they anneal properly with these target sites. If you don't know the sequence here on the purple, the enolase strand here, there's no way you could find the primers that would specifically anneal to this right spot. So you have to know a little bit about the sequence that you're amplifying. The second limitation is contamination. The beautiful thing about PCR is that it can amplify small amounts of DNA into large amounts of DNA. But because of that, if there's any contamination present, you could amplify the wrong target. And because of that, contamination is a problem. You may amplify your gene of interest, enolase in this case, but you may also amplify the wrong gene. So you have to be very careful when you're doing PCR to not have this contamination problem. The other problem is Taq polymerase itself. It's an essential enzyme because it survives at 95 degrees, but Taq polymerase can't proofread. Our DNA polymerases in our cells can proofread. They'll make DNA, like here, and then they'll scan it back to make sure they made it correctly. Taq polymerase cannot do that. So because of that, you can get some errors with PCR using Taq polymerase. Today, though, we do have some heat-stable enzymes that aren't Taq, but they're like Taq, that are heat-stable, and they also have proofreading capabilities. But they're more expensive, so people usually still stick with Taq. Early on, this has been overcome a little bit, but early on, there was a 2,000 base pair limitation If you wanted to amplify something larger than 2,000 base pairs, it was difficult to do. Some advances in recent years have allowed us to amplify much larger targets. Now, PCR has revolutionized a lot of industries, and we use it in a lot of different ways. And your book talks about some of these ways, and you could probably look online for uses of PCR. But you should know a few examples of how we use PCR today. Okay, that's all I have for this podcast. If you have any questions at all, please let me know. If not, I'll see you in class. Bye.